one, really large portion of their ATRT working group. I think ATRT really has served as a galvanizing framework for continual development around the work that we do within CBTN and in partnership with PNOC. And a key part of that is Ashley's co-chair, which I'm happy to introduce Eric, who will talk about the other half of the work that's been done at ATRT.
Great. Well, thank you. It's great to be here. And, it's great to see the avatar that I have. My then 6-year-old son took that picture, and it's a few years out of date, but I think he can continue to get the credit. So it's a good picture to have. Let's see. So it's really great to be here to have a chance to talk about how we're actually using the CBTN data sets from the Pediatric Brain Tumor Atlas to inform translational studies in this atypical teratoid rhabdoid tumor that Ashley told you about. And so that's the goal of this talk, is to kind of tell you how we're actually using this incredible resource to actually advance new therapies for patients with ATRT. So I don't have any disclosures other than using non-FDA approved drugs, which is actually kind of the whole point of this, is to find new drugs that we can then apply to patients with ATRT. So a little summary of what you heard yesterday. Mark, who's a research technician in our lab, showed you that, that when you look at the CBTN data set, you can see that biomarkers of MAP kinase pathway are upregulated in ATRT, and actually upregulated at the level that you would see in low-grade glioma, even higher than that. So low-grade glioma, for example, has activation of the MAP kinase pathway. That's what drives that tumor. ATRT has even more if you're using Sprouty as sort of a pure biomarker predicting activity of that. And then it's been something that we've been curious about and wanting to pursue for a while. And then mirdametinib is the best in class for getting into the brain, and in fact, as a single agent, in a very aggressive ATRT model, extends survival. So now our task, as Mark mentioned, is to find a drug that works along with mirdametinib and then advance that to the ATRT clinical trial that Ashley mentioned is one of the arms. So that's kind of what the purpose of this talk is it's not just our lab doing this. We have about monthly meetings with people from all over the world who will provide their input. Some people part of PNOC, some people outside of PNOC. So it's actually a really open thing. Anyone can join, anyone can bring their ideas, and we'll discuss and then see whether we can find a partner in pharma to work with us. So one such pharma partner who actually approached us is Lantern Pharma, and they have a drug called LP-184, which is a brain-penetrant alkylator, and it's currently in phase 1 clinical trials in adults with high-grade glioma. And MTD has not been reached in that clinical trial, I just heard recently, so that means the maximum tolerated dose keeps going up, and the patients are tolerating it fairly well as a single agent. So it gets into the brain fairly well. It's not perfect, but it's about the same as temozolomide, which is a drug that we use for high-grade gliomas. And then it gets activated into its active form and damages DNA. But one of the interesting things is that either tumors or drugs that impair DNA repair could enhance the sensitivity to this drug. So what I mean by that is that if you block some of the members of this family, like ERCC or PARP, you may be able to accentuate the activity of the drug. And so the drug company brought this to our attention, that there's a drug called spironolactone that's been around for like 1 million years. It's been given to like hundreds of millions of patients. It's a diuretic. We give it to NICU babies. We give it to 80-year-olds. It's a potassium-sparing diuretic. And it turns out that this drug actually binds to, not our work, other people's work, binds to one of the key repair proteins and degrades it. Basically takes it and puts it into the garbage disposal unit of the cell and trashes it. This is called a PROTAC. And pharma companies are designing novel PROTACs, but here's something that was actually found that we know is safe to give to kids that takes ERCC3 or XPB, xeroderma pigmentosa B, and trashes it. And this is what that sort of looks like. It's part of the nucleotide excision repair program. And when we look at this in our preclinical settings, a couple of things to think about. First, the CBTN data set tells us that ERCC3 is actually really highly expressed in the tumors that we care about. They kill a lot of kids. ATRT, diffuse midline glioma, high-grade glioma, medulla. So this gets our attention as well. Can we take this away from those tumors and accentuate the effect of LP-184, or maybe even other chemotherapies like carboplatin? So this is really exciting to see the CBTN data set telling us as a beacon that these tumors could potentially be really susceptible to spironolactone. And when you use spironolactone in vitro and in vivo, you can see that it basically trashes ERCC3, whether it's in ATRT, in vitro at the top right, or in vivo in a flank tumor model. You can see that you significantly decrease ERCC3. And also D283, which is a medulloblastoma series, D341, D425, same story. And in the DMG at the bottom right, you can really degrade this. And so this is sort of like the dream PROTAC, right? So you have something that is already super well tolerated. You know, kids and adults of all ages can take it, and yet it takes away something that, and people are not dropping dead from taking spironolactone unless their potassium goes too high, right? So it's like, you know it's safe, and then you could potentially combine it with chemotherapy. And so this is shown here in this slide. Abby Tyndall presented this at a poster session yesterday, but just to highlight that both her work and the work of Anakriti Bhargava doing the DMG work shows that when you combine spironolactone with LP-184, you really juice up the cell death or apoptosis. And most importantly, you can see that it really enhances the survival of mice with a very aggressive ATRT model. So spironolactone by itself is about the same as control. This is a pilot experiment. But when you add spironolactone and LP-184, you're really improving survival dramatically. So more to come on this, but this is data that we're sharing with the drug company on a monthly or even weekly basis. So they already have a planned phase 1 in children that's going to be starting, Ashley and I just found out, through the Poetic Consortium. So it's going to be a phase 1. We've been talking to them a little bit, that will first escalate the dose of LP-184 based on the adult data, and then add spironolactone on top of it, because I told them my enthusiasm as a physician scientist for LP-184 will be dramatically increased if you do your trial including spironolactone based on this data, where what I don't show you is that LP-184 by itself didn't hit the target in the brain, didn't get enough probably into the brain at a safe level for the mice to actually improve survival. It worked great in the flank tumors, but it didn't get into the brain, we think, enough. But by lowering the needed dose with spironolactone, you're able to actually get where you need to go and get improved survival in the mouse model. So 2 sort of early examples of how CBTN is really informing our preclinical work that will hopefully inform clinical trials. Not theoretical, it's actually happening. So to highlight the part that Ashley mentioned about the first arm of the ATRT clinical trial, we've been interested in this thing called the integrated stress response for a while, and particularly how mTOR interacts with stress. So it turns out that fast-growing cancer cells, and particularly ATRT, this again is CBTN data, express a lot of something called ATF4, which is a transcription factor that's turned on when cells are stressed. And you can be stressed by a lot of things if you're a cell or a person, but it turns out that this is like evolutionarily very conserved, all the way down to yeast. When you're starving, you access this ATF4, and it leads to either an adaptive response where you survive, or if it's prolonged, you go over and you die, oftentimes from apoptosis. So are there levers that we can pull to either stress the cells so much that they go over into dying, or that we block the stress response and then exacerbate the ability of chemotherapy to kill the cells? So we can tell you that ATRT have epigenetic stress because it's an epigenetically driven tumor. We can tell you that oftentimes in the brain, but not in culture, the cells are in a nutrient deficient environment and are stressed. And we can also tell you that because they're growing fast, they have replicative stress. And because they're making lots and lots of proteins, they have endoplasmic reticulum or ER stress. So there's a lot of stress, and that's what's shown here in this graph that ATF4, which is the key readout, is much higher there than it is in low-grade glioma. And mTOR feeds into this. So we've been thinking about mTOR for ATRT for a while and published several papers on it. And the newest sort of best mTOR inhibitor is something called paxalosib that gets into the brain very well. And is currently in development for DMG and in clinical trials for diffuse midline glioma. And we have had some early success with paxalosib, which is I'm not showing you here. And as part of our monthly sort of meetings, international meetings, our colleagues at Princess Maxima in the Netherlands said, hey, we're finding really good results with gemcitabine. Maybe you should consider adding gemcitabine and paxalosib together. And this was great. This was before they published their paper. They're sharing their unpublished data. And that's the whole point of this, is that we're all sort of mad scientists in this together to help patients. And that's what Ashley and the PNOC people have been able to do, is put together this working groups where people both inside and outside of PNOC are able to contribute to try to move things forward. And so they proposed gemcitabine. They just published this paper showing gemcitabine as a single agent works well in ATRT, extending survival. It's this Kaplan-Meier curve show. And so, we wanted to ask, could paxalosib or Pax here and gemcitabine work together? And in the blot on the left, you can see that there's decreased mTORs measured by phospho-AKT and phospho-S6 with paxalosib. But most importantly, in the blot on the right, and this is still early days, it seems like gemcitabine increases the expression of ATF4 a little bit, increasing the stress. And, this is one of those funny things is we think we know how gemcitabine works, but I'm not really sure we know how gemcitabine works, because it could very well be interacting with aspects of translation and mRNA production that is very different from what people traditionally think of as how this drug works. And so more to come on that. But the combination of Pax and Gem seems to shut down ATF4, suggesting that really what we're doing is blocking this adaptive response. And how does that work out in killing the cells, which is what we really want? And this plot shows you measures of apoptosis or cell death, that you can get vastly increased apoptosis in ATRT by combining paxalosib and gemcitabine. And this is Rijal Dangi's work down there. She's just joined our lab very recently, but has actually been an Alex Lemonade Sand summer student in our lab before. So it's perfect. So she already knows all the things to do and just stepped right in, picked up a pipette, and started doing Western blots. This is some of her data here. And then Tyler Finley, who's now a graduate student at Johns Hopkins, has been working on this project for several years and showed that you can really get excellent synergy. Red is very synergistic, which means 1 + 1 = 3 for this. And so areas of synergy that are actually using picomolar concentrations of gemcitabine, so really, really low concentrations of gemcitabine and paxalosib doses that are achievable in the brain. So sort of the model we're working on is that gemcitabine is really going to kick off the stress response by further stressing the cells. And the paxalosib may very well block some of those stress responses, forcing the cells away from survival and more towards death. And where the real money is in the mouse experiments, which is where Tyler spent most of his time, and Ashley showed some of this data. But this is imaging where you're using firefly luciferase to actually make the tumor cells glow through the mouse skull, and you can actually measure the growth of the tumor cells this way. And you can see that if you're a mouse, you really want it to be in the combination because the tumors are much smaller in terms of their brightness there. And the survival curve, the ones that drop down like a stepping stone, you don't want to be in that group because that means those mice had to be sacrificed or died from their tumor. Paxalosib does a little bit, and in 2 different models, and we now have a 3rd model as well, of tumors growing in the brain, you can see that you really want it to be in the combination group because the BT37, which is the tyrosinase group, those mice survived vastly longer than anything we've ever seen before. And CHLA6, which is a very aggressive model, the combination also outperformed. And so this is really like, as someone who would be enrolling patients on this clinical trial, when I see data like this, it gets me really excited. No offense to Mark, but the single agent mirdametinib data where you increase survival a little bit tells us there's a signal there, but that's not enough to launch 1,000 ships and get a lot of people excited about a clinical trial. But in my mind, seeing something like this where you're really able to actually hugely extend survival with something that's generally not toxic to the mice is pretty exciting. So in conclusion, the CBTN data sets really are, I'm going to have to make sure I don't mangle it, really represent an unparalleled resource. And I was talking to Jay, talking to Adam. I remember the early days of this when I was a CHOP. Of course, Jay was taking care of patients. I was a resident in the ICU. And I also remember visiting Adam when he was like, oh, we're building this great resource. And this, I don't know, probably 10 years ago. And you showed me the walk, the -80 freezer with the robots and everything. And it was really exciting. And to see it coming to fruition and actually able to help us inform clinical trials that are going to help potentially help real patients is really exciting. And I just showed you 3 examples. There's a lot more to come because we are just beginning to scratch the surface. This is me, not a bioinformatician, looking through the CBTN data set and finding these things. So I'm sure there's a lot more, and who knows, maybe AI will make this a faster process. But, we showed MAP kinase activation is important, building on our prior work. And, ERCC3, really highly expressed in the bad tumors. We now have a tool that we can take this away. That should launch additional clinical trials. The company is super interested. I think that this is something that's going to happen. And that's also a benefit from CBTN data sets. And then the ATF4 activation really is a beacon telling us that pathway, the integrated stress response, could be something that if we can modulate that, we could really potentially help improve survival, and that's a vulnerability in these patients. Okay, with that, I'll stop.
Pathway activation with the well-known deletion of the INI1 gene on chromosome 22. I mean, what is the connection?
So your question was, do you think mTOR is related to the INI1 loss? And so we actually looked at this, and the way we got interested in mTOR was that it seems like the reason why ATRTs occur, and they occur in babies primarily, is that it's a stem cell that's gone wrong, and there are key stem cell factors that ATRT continues to express, including something called Lin28, which has the moniker of my favorite gene. Lin28 is one of these Yamanaka factors that can turn a normal cell into any other cell. And Lin28 actually helps to regulate mTOR. So what we got interested in was we found Lin28 was highly expressed in ATRT, probably because the cells are stuck at the stem cell state, which makes them so difficult to get rid of. And Lin28 regulates mTOR as well as MAP kinase. And so that's how these 2 pathways became prominent in our minds, coming out of the original finding of Lin28. So Lin28 is something that is potentially regulated by INI1, but it's probably more related to the differentiation block, that losing INI1 keeps those cells stuck in the stem cell state and doesn't let them out. Hopefully I answered your question.
INI1 would be only a secondary element that we use to identify the tumor?
It's a primary element, but it's something that blocks them from progressing. And so there are other drugs that we're currently exploring that I don't have time to tell you about that actually use epigenetics to force the cells out of their stem cell state and to differentiate them to the point where they can actually sort of mature properly and then downregulate all of those stem cell factors.
Okay, thank you.
My question is about the first story you told about this spirulactone. You referred to that as a PROTAC. Was it actually developed as a PROTAC with like the linking E3 ligase, or does it just happen to degrade ERCC3? And if so, do you know the mechanism of the degradation? Or
so this is a great question. It was never developed as a PROTAC. It was developed as a diuretic. It's been around probably since the 1960s, something like that, maybe even the 1950s. So I challenge people who think they know how drugs work to humble themselves, because I think we really have no idea. We've seen time and again that drugs like thalidomide that everyone thinks work one way actually work a totally different way. That's another example of a drug that was found in the last 5 years to actually degrade a protein, basically the transcription factor Icarus. And so, we give all these drugs to people and we think we know how they work. And this is actually very relevant for AI, where, some people are like, oh, we'll just teach the AI all the different drugs and then it'll solve all our problems for us. But that assumes that the data that we're putting in is actually correct and we know how the drugs work. I don't think we know how a lot of the drugs work. And so this was just found in a screen, not by us, by another group that was basically like, how do we restore platinum sensitivity in platinum-resistant cancers, particularly bladder cancer? So they screened, I don't know, 5,000 or something FDA-approved drugs, and spironolactone was by far the winner. It's probably not specific to LP184. Something that we can talk about as a group is whether we want to actually think about doing a clinical trial for recurrent patients where we give them spironolactone and some drug that maybe they've become resistant to see if we can restore the sensitivity, because that's like a pretty low-lying fruit question to ask. It's not something that has to be LP184 specific.
Yeah.
But it's fascinating that they found that this drug, which millions and millions of people have taken, takes away for 4 to 6 hours a key DNA repair protein. People have not been dropping dead because of it. I don't think there's ever been a cancer risk associated with taking spironolactone. So really interesting stuff.
Thank you.